Here are some series of questions on plant for the AQA biology. So it talks about tissues, organs, and their functions. Just some series of questions on plant. Now, first they said a plant is a root is a plant organ, and plant roots contain many types of tissue. What is a tissue? A tissue is a group of same or similar cells that carry out a particular function. So that's it. Now, tissue in the tip of a plant root contains stem cells. Now, stem cells can differentiate into any type of cell, yes. Name the type of tissue in plants that contain stem cell. These are the meristematic tissues, so meristems. Now, in the past, many drugs are extracted from plants. Aspirin is a painkiller. Which plant does aspirin originate? That's the willow plant. Um, Now, scientists have extracted chemical A from a deadly nightshade plant. Chemical A can be used as a painkiller. Now, table 1 shows information about which chemical A, where chemical A is found. Now, we have root, leaves, and berries, and we have 1.3, 1.2, and 0.7 mass of chemical A in 100 gram of plant tissues. Now, they say the scientists usually extract chemical A from the berries of the deadly nightshade plant. So, suggest one reason why berries are used instead of leaves or root. Now, because if you use the leaves will definitely damage the plant. If you cut it off, then the plant will not be able to photosynthesize. So, berries can easily be extracted and it will harm the plant. So, easy access. Can easily extract chemicals, and berries will not harm the plant other than where you remove the root and the leaves. Now, we have a deadly nightshade plant has chlorosis and yellow leaves. Now, chlorosis is caused by lack of magnesium ions. You should know that. Now, because magnesium is used to make what? It's used to make chlorophyll, and chlorophyll gives the plant its green color. Now, they say the mass of chemical A found in leaves of the plant is 60% of the mass shown in table 1. Okay, so calculate the mass of chemical A in 200 gram of a leaf with chlorosis. So you should go back to your table. This is 100 gram. In the leaf, you have 1.2. So 1.2 is equal to 100 grams. So for 200 grams, you have 1.2 times 2. We give you for 200 grams, which would be 2.4. Now, we are now told that the percentage of this chemical A in leaves with chlorosis is just 60% of this 100 grams. So you find out that 60 over it, your answer will be 60 over 100 times 2.4. Whatever you get, they say we should convert it to milligram. So from gram to milligram, you multiply by 1000. So this is what you have. So 2 times 1.2 give you 2.4. So 2.4 times 0.6. This 0.6 is gotten from this percentage, which is 60 divided by 100. Then it will give you 1.44. If you want to convert it to milligram, so this is in gram to milligram, you multiply by 1000. That's all. Now they say name one reason why the leaves of a deadly nightshade plant have chlorosis. I think I've explained it here, which is lack of magnesium. So lack of magnesium lead to lack of chlorophyll. Chemical A has not been tested in large-scale clinical trials in the UK. It is important for drugs to be tested in clinical trials before the drugs are approved for use in public places. Why? You need to check the toxicity, you need to check the dosage, you need to check if it's safe, you need to see the side effect, also to check if the drugs will actually work. That's why you have to do clinical trials. Now, there are many online reports making claims about the effect of chemical A. Some of these reports are biased. Why will a report be biased? Any report can be biased because, in this case, making claims, online report, possibly for financial gains, for your competitor want to have an unfair advantage, also for financial losses. That's it. Now, how can scientists be sure that claims about the new drugs are valid? To be sure, you need to do peer review. So peer review is the answer. Simple. Now, figure 9 shows a cross-section of a leaf. When given this type of question, first you should try and label the part. This is the upper epidermis. This is the palisade mesophyll layers. This is the spongy. mesophyll layers. You have the guard cells here that controls the opening and closing of the stomata. Now, they say which cell is the most transparent? It is A that is the most transparent, which is the upper layer. Now, which cell structure in a leaf cell is not found in a root hair cell? I think that should be the chloroplast. Now, take note, the chloroplast is a cell structure because the chlorophyll is made by the chloroplast. Now, name the cell in a leaf that controls the rate of water loss. That's your guard cells. No, sorry. Yes, the guard cells. That's the answer. Now, water is taken in by the roots, transported up the plant, and lost from the leaf. What scientific name is that? That's the transpiration. Loss of water is transpiration, but the movement is referred to as transpiration stream. That's how you transpiration stream or transpiration pool. Now, which change would decrease the rate of water loss from a plant tissue? Tick one box. Now, the factors that affect it: light intensity. Increase in light intensity will increase the rate of water loss. Increase in the number of stomata will also increase the rate of water loss. Increase in temperature, increase the rate of water loss, but increase in humidity will decrease it, because humidity is the amount of water in the atmosphere. So if the amount of water in the atmosphere is high, there will be no water will not be able to diffuse out of the leaf because it cannot move from low concentration to high concentration. So the answer should be increase in humidity. Now, they say compare the structure and function of xylem and phloem. Two things. You should compare the structure. Then secondly, you look at the function. Now, based on structure, you find out and look at the indicative content. Based on structure, xylem is made up of dead cells, while phloem is living cells. Phloem have end walls, while xylem do not. They don't have end walls. They have hollow tubes. So they have hollow tubes because they are dead cells. There is no cytoplasm. But phloem are living, so they have cytoplasm. Xylem have lignin. Phloem does not. Now, when it comes to function, find out that xylem help to transport water and mineral ions, while phloem help to transport sugar. Specifically, it transports sucrose and amino acid. Phloem help in translocation, while xylem involve in transpiration. Phloem is bidirectional movement. That means material can move in any direction, up and down, while xylem is unidirectional. That means it moves in just one direction, upward. So that's it. If you can explain it with that content, you get your 6 marks. Now, we have another question here. They said, water moves from a plant to the atmosphere through the leaves. Yes. How is the volume of water lost from the leaves controlled? Now, it's controlled by guard cells because guard cells control the opening and closing of the stomata. So that helps to control the number or the volume of water loss. Sorry. Now, describe the transport of water through the plant from the roots to the atmosphere. How is water transported from roots to the atmosphere? So water is transported in the xylem. uh, Water evaporates from the leaves through the stomata. So that will create a transpiration what stream. This will create a transpiration stream. So take note of this, please. Now, a student investigated the volume of water lost from two plants of different species. Both plants were kept together. Now, sorry, let me come. This is transport, not absorption of water. So do not make that mistake by saying osmosis. No. Now, water move through the transpiration stream through the xylem to the leaves. As you get to the leaves, you evaporate. So it will now form a continuous movement. So take note about that, please. Now, a student investigated the volume of water loss from two plants of different species. Both plants were kept together, and this is shown in figure 5, plant A, plant B. Okay, let's see the question now. They say, suggest one reason for the difference in the rate of water loss from the two plants in the first 2.5 hours. So in the first 2.5 hours, which is somewhere here, 2.5, what is the reason for the difference? Obviously, it can be that plant A have more stomata, so it tend to lose more water. Simple. Now, both plants were moved to a different place, at 2.5 hours. Now, calculate the rate of water loss per hour in plant B from 2.5 hours to 3 hours. So if you go back from 2.5 to 3 hours, so we are looking at from this point to this point. Now from those 2 points, sorry, this point to this point. Now the time difference here is like 2.5 to 3, that's like 30 minutes. And if it's 30 minutes, they want us to give the answer in hours. So this is like 0.5 hours is the same thing as 30 minutes. If you do 0.5 times 60, same thing as 30 minutes, 0.5 hours. So what is the rate of water loss from 2.5 to 3? So from 2.5 to 3, at 2.5 it is 2. Now at 3 here it is this is 8, so this is like 7.2. So you have, so the rate of water loss from 2.5 to 3 will be 7.2 minus 2 here, which is this starting, where it start to this point. So if you do that, you now have 5.2. Now to calculate the rate, it's now simple. So if you have your 5.2, it will be 5.2 divide by, because you are looking at per hour, divide by 0.5, which will now give you like I think 10.4 thereabout. So 10.4 centimeter cube per hour. That should be the answer. Let me see. Yes, we have 100 divided by 10.4. To two significant figure is not up to 5, so you leave it as 10. You cannot approximate. Now, suggest 2 reasons why the rate of water loss in both plants change after 2.5 hours. It's simple. It can change because you took it. They say it was taken to what? A new environment. They said, suggest one reason. Where is that statement? Both plants were moved to a different place. So why would there be a change? It's because one, the new place might be warmer, so temperature affected. The new place might have higher light intensity, which affect it. It might have less humidity, which will increase it. It may be windier, which may increase it. So these are factors that affect rate of transpiration. So thank you. I'll see you in the next aspect that I'll be looking at.